Good morning students. In previous video, in mechanism of hearing part 1 video, we discussed about how the middle ear acts as a sound amplifier. Like whatever sound comes in the middle ear is amplified 22 times before entering into the inner ear. The main thing I told you 2 5 marks, impedance matching and tympanic reflex. Why this 22 times more amplification is needed? Because the inner ear is rich with fluid, whereas whatever sound comes is in air medium. So there is a medium change. And in today's video, I am going to discuss about this, what is the fluid present in this inner ear, about the inner ear and how the action potential is being generated in the inner ear. Now you can see here, this is the structure of the inner ear. In general, inner ear is nothing but it is made up of bony labyrinth. Within the bony labyrinth, there is a membranous labyrinth. This bony labyrinth includes, this is cochlea and this is semicircular canals. So we know there are 3 semicircular canals. You can see here there are 3 semicircular canals attached at right angles to each other, 90 degrees. This is utricle and saccule, and these 3 semicircular canals, utricle, saccule is one part, and this cochlea which contains 2.5 turns is also inner ear. The semicircular canals is also inner ear, and I am not going to concentrate on this. The function of this semicircular canal, utricle, saccule is equilibrium, all those things which I am not going to discuss in today's video. I am going to concentrate on this cochlea, which is a membranous labyrinth which is present within the bony labyrinth and the inner ear. And the function of cochlea we all know, hearing. As you see here, cochlea contains 2.5 turns. Now what I want to tell here is, this cochlea which is having 2.5 turns, you can see here some 3 membranes or circles. These are 3 are scala. So scala vestibula, scala media, scala tympani. How this occurs, I am going to explain. Imagine, for example, this is cochlea. See here, 2.5 turns. Imagine a snail, simple. So imagine this as a snail. The cochlea is in the form of snail which is having 2.5 turns. Now I am taking the snail which is coiled, and I am making the coil into straight tube, simple. This coiled tube, I am taking it and making it as a straight tube. Yeah, this coiled tube, I am making it straight. Now what happens, as I mentioned here, it will be having 3 layers, simple. So the top one will be scala vestibula, the middle one will be scala media, the lowest one will be scala tympani. This both red colors is the inner ear. The inner ear contains semicircular canal, utricle, saccule, which is concerned with, so what could be this nerve? Vestibular division of vestibulocochlear nerve, and this nerve will be cochlear nerve. And now I am going to explain how this fluid movements takes place in the scala, vestibular, media, tympani, and how the electrical impulse is transmitted into the cochlear nerve, or how the cochlear nerve is stimulated, to make it simple. Let me enter into I am just going to take this alone. Yeah. So like this, scala, vestibular, media, tympani, this entire cochlea 2.5 turns it is there. For understanding sake, I am taking one scala, vestibular, media, tympani, and of course you know this. This is oval window. This will be round window. Yeah, this is scala vestibule. This is scala media. This is scala tympani. Please note it down. 3 scala. I again repeat. The top one is scala vestibule. How to remember? Oval window. OW, OV we will put. So oval window. So V for V. So top one is scala vestibule. Middle, M for middle, M for media. So make it easy. Oval window, OV. Oval window is top. So the oval window is the one which is striking the scala vestibule, V. So V for V. The top one is scala vestibule. M for middle. The middle one is scala media, M for M. The lower one is scala tympani. So we are having 3 scala: scala vestibule, scala media, scala tympani. So they have to be divided. They each have some type of fluid. So they are divided by 2 membranes. Again note it down. Yeah, this membrane is Reisner's membrane. This one is basilar membrane. This is Reisner's membrane. 2 membranes. So the Reisner's membrane is the one which separates the scala vestibula and the scala media. the basilar membrane, the top, the blue color you can see here, this blue color. This is the basilar membrane which divides the down scala tympani and scala media. Now next, what fluid is present in each scala? Scala vestibulae contain perilim, scala tympani contain perilim, and scala media contain endolymph. MCQ, very important, and you can note it down. And also you can note this, endolymph is rich in potassium, less in sodium. It is reverse in perilymph. I again repeat, scala media is rich in endolymph and endolymph is rich in potassium, low in sodium. Please note it down. We are going step by step. We finished the inner ear, 2 nerves. I am now entering into this salon. You see here. Now you can pause the video and try to answer. What is this blue color? Yeah, yes, it is basilar membrane. And above the basilar membrane, you can see these 2 rods. So this is nothing but rods of Corti. Note it down. These 2, which is resting on the basilar. Otherwise, there are 2 rod shaped structures which rests on the basilar membrane. You can see here 2 rods. They are called rods of Corti. And their space here is obviously tunnel shape. You can see here. This is called as tunnel of Corti. So these 2 black ones, these are rods of Corti, and this space almost V shape or inverted V shape. This is called tunnel. Tunnel means space. Tunnel of Corti. Now I want you to concentrate a little bit more. On either side of the rod of Corti, you can see 3. These are called as outer air cells. Outer air cells, almost I have drawn 3. And this side also there is 1. This is called as inner air cells. Inner air cells. So on either side of the rod of Corti, I am having outside, outer air cells, 3, almost 3 I have drawn. Why I have drawn 3 outer air cells, 1 inner air cells? The ratio is like that. For example, if you are having 1500 inner air cells, means there will be around 4000 to 5000 outer air cells. Almost like 3:1, not exact, but outer air cells are more in number, but inner air cells are very less in number. But I am going to explain. In case if they ask which air cells are very important in hearing, both are important, but which is responsible for main hearing, it is inner air cells. Note it down. Inner air cells are more very important, and inner air cells are the one which send the afferents. Inner air cells are the one which send the afferents to the auditory nerve and which is responsible for hearing. Or 95% of hearing is because of inner air cells, or 95% of the nerve signals which are transmitted are because of inner air cells. Now next question. Then what is the function? I will write here. Inner air cells mainly hearing, discrimination, amplitude, all. Outer air cells, they mainly modulate, like modify, or they have little bit control. Outer air cells are the one, for example, if too much sound comes, maybe this outer air cells helps in decreasing that sound. Or otherwise, one more example, various frequencies or sounds are surrounding me, I want to hear to a specific frequency. There again, outer air cells helps. So I am just telling the difference between inner air cells and outer air cells. Both are lying above the basilar membrane on either side of the tunnel of Corti. For one inner air cell, there are approximately 3 outer air cells. Inner air cells are the main, in fact I can tell receptors. Inner air cells are the main receptors for hearing, but outer air cells are also important. They mainly modify. This green color you can see, they are called supporting cells. Supporting cells like Dieter cells, Hansen cells. So all these are supporting cells which supports the air cells. And you can see here black color here, please note it down, reticular lamina. So now again I will ask you, what is reticular lamina? The exact at the top of the air cells, there is a lamina. The lamina present on top of outer air cell as well as top of inner air cell as well as top of tunnel of Corti. There is a lamina like structure that is called reticular lamina. Note it down, reticular lamina. And these are called stereocilia. Cilia, you know, no, cilia, finger like projections. The air cells above are having cilia like structures. You know the function of cilia, moving to and fro motion. So you can see here 3 cilia, cilia, cilia. Inner air cells are also having cilia. This black color is reticular lamina. So below the reticular lamina is air cell, above the reticular lamina is stereocilia. But remember, the stereocilia is being obstructed. In fact, we can say the stereocilia ends at a blue structure. You can even try to pause the video and answer, and anyone tell me what is this structure name. That is a membrane which we call it as tectorial membrane. The stereocilia strikes the membrane which is called tectorial membrane. So the stereocilia ends at tectorial membrane. The stereocilia begins at reticular lamina. The outer air cells, inner air cells ends at reticular lamina. The air cells are supported by supporting cells. The rods of Corti begin from basilar membrane. The space between the two rods of Corti is called tunnel of Corti. And whatever I explained so far, this entire structure is called as organ of Corti. In vague term, we can tell organ of Corti can be considered as receptor for hearing. Obviously, organ of Corti is inner ear. If specific, air cells can be considered as receptors for hearing. Now coming to the actual mechanism, I will just give a brief idea. I already told this entire inner ear is filled with fluid, both perilymph as well as endolymph. And you can see here, this is the 8th nerve and this is coming from inner air cell. See here. So the inner air cell is sending the afferents to the, note it down. That's why I told inner air cells are very important. Inner air cells are sending the afferents to the peripheral process of spiral ganglion, and this continues as action potential in the cochlear nerve, which I will be explaining how the action potential is being generated. Then what is the role of outer air cell? I already told outer air cells modifies the sound. For example, if too much amplitude sound is coming, what happens? Another thing occurs here. We call this as olivocochlear bundle. What it does? It gives efferent. So from the olivocochlear bundle, efferents come to the outer air cells, and so that some modification of the nerve signals or the change in the potential occurs. So inner air cells itself is responsible for generating action potential in the cochlear nerve. Outer air cells can modify that action potential. It can either increase the nerve signals or decrease the nerve signals. And from the inner air cells, afferents goes to the peripheral process of the spiral ganglion. From the olivocochlear bundle, efferents comes to the outer air cell. So this is efferent, this is afferent. So that also we can note it down. Now this is rich in endolymph, this is rich in perilymph, which is producing this fluid. We have in general, we call that as stria vascularis. So stria vascularis is the one which is producing this both perilymph, endolymph, and endolymph is rich in potassium, low in sodium, whereas perilymph is low in potassium. That also I want you people to note it down. Now let me explain how this movement of air cells is going to generate action potential. So hope this video was useful to you. We will meet in next class, mechanism of hearing part 3. Please share this video to our students community as exam is also nearing. This is actually expected essay. Okay, we will meet in next video. Thank you. Have a nice day.